So I'm really happy to be here. Thank you for the invitation, and thank you for accepting Agriodor to this e-niche group. So I'm NLA-PIC. I'm Estonian, but I did my PhD studies in France, and since almost 20 years, I live in France. I founded a company in 2019 that's called Agriodor, and it's based on semiochemicals, chemicals. So we are based in Rennes. It's a nice region in Brittany. We are about 35 people, but it changed. I think we'll be 39 soon. And we are a private laboratory in applied chemical ecology for agriculture. And we're developing biosolutions. So it's not biostimulants, but biosolutions based on pheromones and allomones for pest control. So what we do, when we talk with scientists like you here in the audience, we talk about allomones and pheromones, but normal people don't understand that. So we talk also about attractants and repellents. So what it's known is, as you know, everybody knows that lots of insects use plant odors to locate their food resources and find the oviposition sites. So we take advantage of this relationship that insects have with their plants, and we create products that change the olfactory environment of insects. So it's a fancy way to say that we put odors to protect crops. So it's a nice idea, but there are lots of challenges to do that. So we had this challenge in 2021, a challenge with a big crop, sugar beet. And it was a big problem in France. In 2021 or in 2020, the neonicotinoids were banned in France, and there's a big sugar production in France. And the neonicotinoids in seed coating were used to control aphids. Aphids, it's not a problem, but the virus that aphid is a vector is a problem, and it causes a illness or a symptom called yellowing disease that brings down the photosynthesis and the sugar yield. So it was a really big problem in France, and the French government said, okay, we banned neonicotinoids, but we launched a big national program for research and innovation to find alternatives to neonicotinoids. So it was launched December 2020, and they asked whether Agriodor, that was a young company, we were founded in 2019, but we're a spin-off of INRADO, so we had some advantages. They asked whether we want to apply, and we applied, and in 2020 winter, before Christmas, we had visitors, and I was on Wikipedia looking up what are aphids. Because before that, I was working only on Lepidoptera, pheromone identification, and the plant-insect interactions, and I discovered aphids. They are polyphagous, they make clones, they have different, they are wingless, and they are with winged, and they change host plants, and it was really complicated. But what we saw, really, it's possible that they are sensible to odors. And so, without knowing aphids, we wrote a proposition and said we could manage, so it was in 2020 winter, before Christmas 2020, we said we can manage aphids with volatile organic compounds, and we said, based on the literature review, we can create a repellent effect, impact on the reproduction, and feeding disruption. So when I did this literature review, what I saw all the time was Tardouri, Tardouri, Tardouri. It's a name. there was some Tarek Tardouri who did his PhD, his postdoc, only on aphids, and he published everything. So my first mission in 2021, to recruit Tarek Tardouri, to have all the information that he had done during his PhD and his postdoc. So he came to Agriodor in April 2021. He looked at a proposition that we did, and he found it's good. And what he proposed is that we work on laboratory to validate that we have really these three effects. We work on microplots, small plots, where we can select plants, select orders, different orders that have an effect, three effects. And further, in white stripes, that will be later in second year. So in the lab, what Tarek did is really he worked on repellent effect. That is, he put aphids on the sugar beet plants, and he put different orders next to the sugar beet plant and the aphids, and he counted how fast the aphids went away and what was the effect. And everything that he did was in a dynamic olfactory system. That is, it's not the air, we sampled all the time the air, and it was not static. We didn't have the saturation. So we put directly in the perspective that we need a concentration that will be effective also in the field. So we needed to know what order at what concentration has an effect on the aphid. So he did a repellent effect. He looked at the reproduction. For the reproduction, he took the sugar beet leaves, put on agar-agar, put all the insects of the same age. It's really fastidious work to have the cohorts of the same age. And he looked at, so it's the reproduction, how many babies or how fast the aphids reproduce with different orders, and with some orders we had the effect more than 35%. And last, he looked also on the feeding behavior. I think he looked also on clip cages, and it's the development. So I didn't show it. And he looked on the feeding behavior, because as I said, the aphids are not the problem, it's the virus that is problem. And the virus transmission occurs when there are pollen ingestion. So he saw that in presence of volatile organic compounds, we have the searching period where the aphid is searching, not ingesting, is increased. And in presence of these volatile organic compounds that we decided to keep, we have less ingestion phase. So it's with electropenetrograms. So I'll be fast. What we saw in the lab, we had a blend. We had several blends, mixes of orders that we decided to test in the field to see whether we have the same effect in the field as in the lab. In the lab, we worked with apters, the wingless aphids, and in the field, we have everything. So how do we take the orders that we identified, we need to take them to the field. For that, we needed to work on formulation. So there is only one slide of it, but we worked on granules because we are working on sugar beet field. And first we started with spraying, because it's the most everybody uses spraying in agriculture. But when you think about sugar beet, sugar beets are really small at the beginning, and it's in the beginning that they need a protection against aphids. So in April to May, sugar beets are really small, and if you do spraying in the sugar beets field, about 90% everything that you spray goes on the ground, it's lost. And also another thing, formulation of volatile organic compounds in microencapsulation, in spraying, it's a challenge. We took the challenge, we did it, it was awful. We didn't succeed, but luckily we worked in parallel, also different formulation, and we worked on granules also. So because we weren't successful in spraying, but we were successful on granules. And what we know, we did some work with farmers. They use granules against slugs. So there are almost the same granules, same properties. You can use a small quad to spread these granules, and we incorporated our volatile organic compounds in these granules, and we put different coatings on the granules so the volatile organic compounds could be kept inside and released in time. What we had is 28 days of stable release, because as I said, when we did lab testing, we measured all the time the concentration that was in the air that has an effect on the aphid behavior. So we were monitoring also in the field what are the granules releasing, and when we go under the threshold of the molecules that have an effect on aphids. So it was about 28 days, and we decided we need to reapply. So this test we did in microplots. In microplots that are about 49 square meters. So these are our people. They were counting aphids. So they were counting aphids on microplots to identify the species, and also if they have wings, if they don't have wings, to have this kind of data, to have an effect, to see what kind of blends have an effect on aphids. So Emma told about the hurdles, about the regulation. So I will give you a small anecdote. Based on this data on microplots with Tarek, we saw we have two different mixtures of orders that have an effect. One order has a blend or mixture of orders had an effect on wingless apter aphids, and there was another, a really strong effect on winged aphids. So we were really, it's nice. We have two different orders. Nice, we have two products. We can, it's a really, scientifically, it's a nice result. Till the next monthly meeting where we were sharing our data with the business development team. And for them, it was a horror case when they say, you don't know what you have done. It will cost 1.5 million for one molecules, and what you are proposing is 5 molecules for wingless and 3 molecules for winged aphids, so forget about it. It's not possible. So what we had to do is really find a compromise, a blend that has effect on aphids that have wings, colonizers, and the apters. So what I want to say is that in science, we can do lots of discoveries, but there's a reality. There is a business development team that says it's not possible, so it wasn't possible. So we tested the data that I show here. So it's the compromise. It's the blend that has an effect on the two types of aphids. And it's the same thing. So we tested also on pig fields. It's not big fields, it's like 1 hectare. So you see on 1 hectare we can't count all the plants. So we had marked plants where we counted every week the aphids. There you have the small tractor with it, with what we can spread the granules. And what we have is, let me see, sorry, up, I think it, what we have is that we can delay. I don't see where I wrote it. That we, what we can do is that we can delay the insecticide use. I can't see where I wrote it. Perhaps I didn't wrote it. Okay, so this product, what we are developing, is not a standalone product because it doesn't kill. It creates an olfactory environment around sugar beet that the aphids don't like. It's based on odors that imitate how do you say, the rosemary and this kind of type of plants, aromatic plants. So aphids don't like these odors. It imitates this odor. So it doesn't kill the insect. Insect goes away. It's deranged by the odor. And this kind of product can be and should be combined with insecticide use. And it delays, for example, the threshold before the first insecticide use about 2 weeks. And it delays the explosion of the population. So it's not killing the population, it's the population management. So as an Agriodor, what I find really fascinating is that we can work on semiochemicals. It's challenging. It's the fun part of our work to do these kind of products. But the big challenge is the formulation, and I think it's often overlooked. We can have an effect in the lab, but we should also, it would be nice if you want to really transfer your knowledge, and so it could be used in the field for crop protection, for example, to know the dosage, at what dosage, and how long these molecules have an effect, and after that we need to formulate it. So this was a showcase of sugar beet. We are working also on whiteflies and thrips and fruit flies, and we are mainly working on alomones, but we are working also with attract and kill strategy to attract with pheromones and to be associated with insecticides for pest management. So thank you for your attention. And if you are interested about Agriodor, or if you have some innovations that you would like to transfer to the field for crop protection, come and see, and we can talk. Thank you.
I am Teun Dekker. I actually represent BioInnovate these days. I quit my academic job and went commercial. I don't know what it means, but it's a very small company. One of the reasons why I actually made a shift is that I love teaching, I love doing research, but I also want to see the impact. And sometimes I miss that a little. We do research in our bubbles, in our silos, and sometimes someone compared scientists or anyone in society really with a lot of ducks in a pond, and they quack very loudly, but no one outside the pond actually registers it, right? So it's a little bit like that. How can we just facilitate? And Inish is a really good position to actually make that connection. And the former two speakers, Emma and Anna, just really highlight already one of the issues, and the regulatory. They're really blocking the innovation that we want. We are smart people here, and we're doing fantastic research, and we come with solutions, and at the end of the day, they're being shelved for a number of reasons, and one of them is regulatory. Okay. So this was a little bit of the abstract that I sent out, but I would like to point out, like, that ENIS has a very important role to fulfill, or anything, initiatives like ENIS, in trying to bridge these silos and make this communication go and make the impact we need. What I miss here is actually the regulatory people that actually, they should be informed. They should be informed because they're bureaucrats. They don't know. So I have a lot of contact with people in Africa, regulatory people, as well in Sweden. And as I once visited the people in Sweden and said, well, this is what we're working on. Thank you for coming. We don't know, just that type of thing, because they need to be informed for legislation to change. Something to say here. Pardon?
Write a paper on this.
Yes, and that's another thing. Indeed, write a paper on this. Jen was saying, and this is true. White papers or policy briefs and things like that, super important. Devote some time to it. We're being paid by society because they believe in us doing something good. But if we can't convey that message, it's lost, right? Okay. Good point. So I'm an avid biker. I love biking. I love Tour de France too. Tour de France. So we're sort of these scientists, and this Carcassonne is a very nice area to bike. And so you think, okay, there's a little bit of hurdles. That's nice, but every etape is actually quite long, and you need to see all the way through. And you even have to Mount Bagnières to get there. But I wanted to. It's really important that we see this whole trajectory. When we talk, when we write grants about what impact it can make, see it through. Oh, yeah. Yeah. Done? Oops. And the regulatory constraints are rightfully being put in place because of the impact our agro-ecosystems have on the environment. I'm talking to the preaching to the choir, so you all know that. They're there, put in place to make sure that we don't poison our environment. That's a good thing, but now the same regulatory rules apply to whatever green products, and we have seen some of them that are put in place. And it still costs 1 to 2 million to register a single compound. And if you're lucky, if it's in the ECHA database, I believe, then it facilitates. But it's an insane amount of time and money, and no small company can do that. So it's actually feeding straight into the big ag company. That's another issue that we need to address here. We are breaking innovation this way. We don't give the small companies a chance, and the small companies lead to big companies. Anyways, the regulatory it's a good illustration. It's really important because in the IPM scheme, you start with cultural, then physical, mechanical, then biological control. In the end, you do chemical. But the farmer doesn't reason that way. They will spray because they have calendar sprays, and this is part of the cycle. If we don't have the regulatory, it's going to be a mess. So nothing bad said about regulatory, but we need to innovate it because it's not fitting the current scheme. So how can we accelerate green innovation? And I would like to I've been researching such a long time and so many different insects, but I really hope to make some impact, and I'll show you some of the impacts at least. We also want to maybe highlight one of the questions we as researchers may want to ask as well: are we doing it right? Is one thing, do you do right experiments and all that, and statistical significance and all that, but are we doing the right thing as well? Because we know that we need a transformational change in agriculture. Our production systems are really terrible, and we may well come up with a chemical ecological solution that just feeds into the same paradigm. Think about it. We're smart people. We can think about these, and impact stretches beyond papers, right? I give you quick 5 waves of what we've been working on. The oak processionary moth in the Netherlands, also a big problem in Germany and the UK. And we started thinking, can we do, even in high trees, mating disruption? It's a moth, so you have pheromones. So we're spraying pheromones. You can control them with BTI and nematodes, and they are being labeled as biological, but of course they have a broad action. I'd like to avoid that. Oak processionary moth is also a health issue, not only defoliation. For the people that don't know, they have urticating hairs. That's really terrible stuff. And they go through under the populations in the it is increasing again across Europe. So we had several phases. Phase 0 to phase 4 were already going from manual applications with a stick to paintballs, manual fills, to making paintballs mechanically and testing a whole bunch of variables with partners in the UK, Netherlands and Germany. The first phase, we could see a clear reduction in the population, really nice. Then, of course, we realized, okay, well, we can't go with a cherry picker in every tree in the Netherlands. And so we went with filling paintballs manually with plastic balls. That was just a terrible job. I think we filled by like 15,000 of them, and we shoot them in the tree. But the results were good. I was, by the way, all over the news because people love to do that, the paintball shooting. And what we call in the Netherlands is cucumber time. There's nothing else in the news, so you will report on this, right? And we saw very nice results there too. Good suppression between, say, 50 and 70, 75% reduction, which is just what you probably will need. Yep. And then we started producing paintballs. There's a lot of research on that, on the formulation as well. So they don't, like, there should be a paintball coming out. I don't know. At some point, should be. But basically, they're little paintballs. We need to have the formulation making sure that it doesn't attack the shell. So there's a lot of development in that, and we succeeded in that. I don't understand why this. Yeah, it's so nice. We were all over the news in the Netherlands. I love that, right? Okay, okay, there you go, and then next is coming out. And people working in the green sector love it. In the Netherlands, they are still asking, when is it going to be on the shelves? I'll tell you why it's not on the shelves. Okay, never mind. This you know the thing. We went to Syngenta because no one can pay the regulatory costs, the dossier, 2 million easily, and the bigger. No, the more specific a product is, the smaller the market, the smaller the margins, the smaller the longer time for return of investment. Syngenta said, is it an ACHI database? No, okay. And I said, well, I know typically after we had 4 talks with them, there's a big company, it's the only one can do it. They said, you know, we typically invest in products that are further down the line. And I was having these balls in my hand and I was thinking like, what? They have other products on the shelf. I'll leave it at that. Second, Splat Verb is another product that was sideways a little bit involved there. It's spruce beetles are a big problem in Europe and they attack the trees, but also when you have felled trees, they cannot be logging them right away oftentimes, and those are perfectly inocular for spread of this pest. So, but if you have the verbenol, that's a pheromone, but if there's a lot of pheromones being released or being oxidized to verbenone, which is a repellent. And if you treat these trees, these felled trees with verbenone, there was at least a test, verbenone, then the idea would be that you reduce the infestation, just like Anna was saying with the aphids. And that's exactly what's happening. You treat the felled trees and you reduce sort of the inocular by 20 days. And that's just about the time maybe to take them out and prevent the trees to provide a new generation, okay? And there we go, Syngenta, same thing. Because who's going to register verbenol? Yeah? And they have other products, okay. And I want to contrast this with 3 very short stories that I have from Africa. So in the EU, we're stuck in molasses when it comes to registration. And in Africa, it's a totally different story, I'll tell you. We work, so this is another beetle, also an Ambrosia beetle that attacks coffee and bores into these, makes galleries, and then, you know, you get these wilting trees, and coffee production is highly affected. Interesting, it's it's a agroforestry setting with lots of different crops, and there's inocular trees there as well that the beetle like. You think, oh, maybe we should cut them, but actually that's a savior for control, because now we can chase the beetle using verbenone out of the coffee trees and into the other trees. We did a lot of experiments there, and consistently we get about 70% reduction in infestation rate. And that makes a huge difference for the farmers, of course. We're talking smallholder farmers. Most of them even haven't had a cup of coffee ever. You know, goes to show. That's not so important. Another story, working with mating disruption. This is a quite simple pheromone, Z914 acetate, and worked with a fantastic team in Iringa area. And no one cares about, full armyworm everyone knows, but the smallholder farmers, capital-wise, not so important, but I thought, let's try this. And they have a lot of human power, and so they started applying mating disruption manually instead of like machines or whatever you have to think. So we worked with this team to apply, and we made a little movie, but there's no time for that. Just the background a little, maize is normally not sprayed because the value is quite low. But because of fall armyworm, solely because of fall armyworm, people start to spray 5 to 7 times on a crop. The margins are very little, and they do it in a way as you see, all right? They have a lot of health issues there, and it's totally underreported. So thinking about impact, if you can change that, that would be fantastic, right? These farmers, these women were weeding there. We asked, are you not afraid for the side effects? They said, yes, but it's a problem for tomorrow, literally. Yeah? Okay, and they spray lots of bad stuff. Here we are applying pheromone, and they love that because it's every 5 meters. It's easy to apply, so it's something like that. Let's see, and there's a little dollop there through which a pheromone is being emitted. I'll skip this one, just quickly showing, compared to the farmer practice, spraying, we did so much better. And I mean, these are farmers. I mean, we worked with 75 farmers in this plot. The other experiment I didn't have time to show with 85 farmers, and it's a massive effort. And the results are very consistent. Also in yield and the number of damaged cobs, which is also important. You get secondary infections with maize, like fusarium and health problems associated with that. So fantastic results. And then the last, quickly, we're still ongoing in Tanzania, doing experiments with Tuta absoluta, huge problem. And seriously, I've been in Africa a lot, but I don't eat tomatoes there. They're being sprayed at least once a week because of Tuta. You don't want to eat it, just little advice, don't do it. Because that's not the nice stuff that they spray either, right? But it's a huge problem because it's a cash crop. It's very important that they're getting sprayed. This is the effect of Tuta if they don't spray, like easily 100% damage if you don't do anything. And I have this little movie. Let's see if it runs now. Come on. Again, it's the same thing. No, I'm not patient. No, I don't see the cursor. The same thing. They spray with a backpack sprayer every week. And this is a nice video because the farmer was actually speaking. And these farmers are not just speaking to your mouth, because it's their living. And they tell you. And this farmer was very doubtful that it would work. And he told literally, like, I started walking in the field and I couldn't believe my eyes. There were no. He hasn't sprayed his tomatoes until like the 3rd or 4th week of harvest. Then Tuta started coming in and then he didn't take his chances. And now he says, well, it's actually disappeared, but I continue. But just because of that, we already cut 12 sprays. And so simple. Okay, and the important take-home message here, you see the captures of males in the treatment plots are much lower. It takes about 3 weeks, and they have to reapply because then the pheromone probably has evaporated from the little wax dollops. This is the point, and I would like to discuss this as well with you, is now I'm working with the regulatory agencies in Tanzania very strongly on mosquito control products, on fall armyworm, Tuta absoluta, the coffee berry borer or the coffee twig borer, and they want this. they're eager, and we're working with the Swedish authorities as well, and with Bill and Melinda Gates and companies, bringing the knowledge together to inform people and start to design regulations that allow the real green products to be accepted. It's unheard of in Europe. So we're making huge, and Bill Gates wanted to get rid of his money, so there's a good moment as well to say, like, I think the biggest impact in sustainable production is if we can change regulatory in a good way. I hope that we will get there with the Tanzanian and the East African community. I'm hopeful for that, but I really wish. We have no time to lose. We've got no time to lose. We need to do something. I think e-niche is a perfect situation. Continue with this beyond this grant, but invite the regulators, invite the politicians. We're stupid, though smart. I mean, we're so smart, but we're working in silos. We need to break these barriers and change regulatory so that we can actually get the green innovation that we want. That's all I want to say.
The title of my presentation is Seaweed Production at BMRS, the good, the bad and the ugly. Okay, so, right. So first of all, where are we? So there's Ireland over there. So we're very far south. We're probably nearly as far south as you can get. We're on this peninsula called the Sheep's Head Peninsula. And that's what it looks like, you know, on a nice day, which normally you can't see it at all with the clouds and the fog and the mist and the wind and the rain. So, and so on, yeah. So a little bit of history. So we were established in 2005. So 20 years this year. We should really have a party or something, you know. So we're an aquaculture company focused on the delivery of marine-based products for animal, human, and environmental applications. So our assets then, we have a state-of-the-art cleaner fish recirculatory aquaculture system in Ireland and in the UK. The UK part of the operation only started a year ago. It's mainly Brexit related because we had a lot of customers in the UK and, you know, the new regulations and import-export was blowing our minds, so it was just easier to set up over there. And so cleaner fish, just in case, does anyone here, hands up, who knows what a cleaner fish is? See some nodding? 2 people. So 3 people? Oh, great. Okay. So cleaner fish they're little fish that primarily eat lice from salmon. So sea lice is probably one of the biggest issues with salmon farming. They're always trying to get rid of lice and they're not allowed to use chemicals anymore. A lot of the seaweed farms in Ireland have organic status. So these little fish, we grow lumpfish and wrasse, and yeah, they eat the lice from the salmon. So, but only when they're small. When they get big, they don't. So the salmon farmers have to keep buying the fish every year, which is handy for us. So we also have a seaweed hatchery. So we're the only, well, not the only, but I guess we're probably the biggest. Yeah. So we supply most of the farms in Ireland, nearly all of them, and we have some customers in Scotland as well. And we have 3 marine sites. So we grow at sea in 3 different bays with organic status. So currently we grow 2 kelps, alaria and saccharina at sea, and on land we are growing spargopsis, and we're trying to grow porphyra as well, which is nori, with the help of Helena, because she's the expert. So Helena now, I'm sorry there's a bit of an overlap between both our presentations, but basically this graph is just showing you that in Europe we're so small potatoes, as we say in Ireland, compared to the rest of the world. You know, you see China there, Indonesia, South Korea and Philippines have the bulk of the production, nearly 35 million tons. And but actually what I learned at the conference that we were both at recently in Canada, they've issues with global warming there and marine heat waves, and you know, there's been a drop in production on the kelp side. But that isn't shown in this graph because I stole this graph from Google. So, okay. So we'll start with the good. So Helena already kind of mentioned all of this. Around 70 to 75% of the world seaweed production is used to make food. And then you've got alginates, carrageenan and agar use 15 to 20%. But the European demand for growing seaweed is growing every year. It's estimated between 5 and 10%. And most of this is this demand is satisfied from Asia, and the European market for seaweed could be worth 9 billion in 2030, with the demand in Europe forecast to rise from 275,000 tonnes in 2019 to 8 million over the period. Actually, Helena had 2050, but what I read on the EU website was 2030. I'm like, oh my God. And creating around 85,000 jobs. Actually, we're involved in a tender for the Commission on where we're going to grow this 8 million tonnes of seaweed, where we're going to put it. Like, you know, it's, you know, so we've created a model of where the best places it can grow in Europe, which is nearly everywhere. But then, you know, like, where are you going to put all the other things? You know, that's over to you, governments. So, but there's loads and loads of new products coming from seaweed all the time. Like from in that 10-year period, there was 2,500 new products containing seaweed introduced. But the price of seaweed is dependent on the processing and subsequent use. So whether it's used for fertilizer or whether it's used for some sort of pharmaceutical, you know. So the bad then is the costs, right? So basically in Europe, 95% of all the seaweed is wild harvested. So if you try and grow your seaweed, the customers think they can pay the same price as if they got it from the wild, which is unrealistic. So here is what we did for the Irish government. We totted up how much it is to set up a farm. And so let's say you had a 100-hectare site in an exposed area, so you've more infrastructure. You're talking about 1.2 million euro just to set it up, right? So then you have to sell your seaweed after that, which, yeah, you know, which is hard, you know. So the ugly, then, this is the bottlenecks. So how do we make this seaweed farming cost effective? So I suppose the issues are, a lot of seaweed farming or seaweed companies like ours, we're all really small, independent farms, you know, that are kind of totally not connected in lots of ways to each other. And you're trying to grow your seaweed. And then you've got massive competition coming in from Asia. Like, we've no shortage of customers who come all the way to where we live, as I showed you on the first slide, all the way to Bantry, southwest of Ireland. Like, you're talking about nearly dirt track roads by the time you know, sheep is all you're going to meet by the end. And, you know, they're like, oh my God, we love your seaweed. That's some of our kelp there in the picture. And it's like, oh, and then they're having a great time, and then we talk about the price, and yeah, you're just kind of going, there's no way we can sell it to you for that. You know, it's nice that you want it, but you're just going to have to pay for it, you know, more than what you're offering. So that's a huge issue for us, and wild harvesting as well. The customers, that's what they think that they can pay for, the farm stuff. So farmed kelp as well. So like, you know, I think we're all growing the wrong thing because everybody grows kelp because it's got the biggest biomass. It's really easy to grow. You've rarely run into issues with it. And but you know, most of the customers around, Helena will, you know, they don't want kelp. They want red seaweeds. They want nori. They want palmaria. They want, or even palmaria is in trouble at the moment. They want other seaweeds that we do not put energy into growing in Europe. Then you have the drying and processing. It's really expensive. So to dry, we can't dry our seaweeds outside in Ireland, you know, apart from that one day that the picture was taken. And, you know, it's very expensive. About 50% of our costs go into drying this kelp, which, so, you know, there has to be other ways to process it. So it's difficult to market as well because there's so many uses, and there's a long value chain. And companies like us who are small, we have a staff of 20 people, we're trying to do it all from the very start to the end, which is absolutely impossible because we're not, if we want to make an extract, say, we're not chemical ecologists, sorry, we're not chemists. I'm sorry to tell you that now. And I'm in the core group. So also the regulations, where's Toon gone? Because it's, yeah, they're so complicated and, you know, well, initially when we started out 20 years ago, there was none, right? It was hilarious because trying to apply for a license, we couldn't. We had to apply for a shellfish license. We're trying to explain that it's not a shellfish. We want to grow, it's a seaweed, but now, yeah, the regulations are quite complicated, and you know they're setting limits on seaweed that, you know, it's how should I say? It's kind of making it really difficult, you know. Like I learned, actually, Helena gave a great talk at the conference in Canada, and she was, one of the take-home messages for me was like, a lot of the products that we eat today or drink or whatever, if they were invented this morning, would never, ever, ever be allowed. Like coffee, for instance, like it's addictive, it gives you the shakes. You know, alcohol is another one. It would never be allowed. You know, it's, you know, but anyway, so seaweed, I guess, is considered relatively new, even though it's been around for a long, long, long time. So also, as mentioned above, you know, we've very few domesticated species. You know, in Ireland we have 600 species and only 13 species are exploited. And so academics out there, like, study some other species, please, and tell us what uses they have, or, you know, you'd never know, one of these species could have the cure for COVID or something. And yeah. So then, this is my final slide. So how do we compete? It's not all doom and gloom, I think. So first of all, I think we need to reduce the production costs. And this can be done if we optimize the farm design and invest in mechanization to reduce labor-intensive practices. Larger sites and integrated sites, like Helena mentioned, integrated multi-trophic aquaculture. One of our sites is beside a big salmon farm, and it's our best producing site every year. It grows faster and it has the biggest output. And, you know, sharing of resources as well, especially in the one area, could be a way to reduce costs. Then new processing methods, some alternatives to drying, like fermentation. You know, there's pickling, there's lots of different things. But you have to get your customers to, because they like getting it dry, so they have to kind of get behind you on the new processing methods. So then emphasize value beyond the production costs. So storytelling, so to showcase the benefits of the environmental and social value to build consumer trust. Also certification schemes that promote environmental and social responsibility, like the ASC, the Marine Stewardship Council Seaweed Standard, and valorising ecosystem services that seaweed farms provide. But I think the third bullet point on developing a European seaweed-based market is most important. And we need to develop products that resonate with European culture, because, okay, there's a lot of products from Asia. We all know about sushi, but we need to develop our own for our continent. We need to emphasize that the seaweed is local. We should emphasize the superfood trend. Everyone knows it's a superfood. So incorporating seaweeds in traditional foods like sausages, pasta, sauces, anything. And also follow Asia's lead by developing kelp-based industrial clusters. They're very good at that everyone works together in the areas of alginates, functional foods, sugar, alcohol, cosmetics, medical materials, you name it. And also because we are very good at creating high-value products, and that's in the pharmaceutical area. So we need to work together with pharma companies who are finding bioactives from seaweeds all the time and creating products out of them. And so we need to work together, basically, as Helena said, create a bridge, work with, academics as well. That's you guys are the people who have the time and the wherewithal to study. We're always about the bottom line and our investors and keeping our customers happy, and actually, growing this seaweed, as soon as you've harvested, the next cycle begins and we're back to the hatchery and everything. So even though we'd love to be able to research more, like someone asked me this morning, like it's your discipline that will help us make better products and compete with other continents. Okay. So I think that's me. So if anyone wants to come visit, we're so far away. Like, I'm always amazed when anyone actually makes the effort to come to the Sheepshead Peninsula. It's really pretty. That goes without saying. And but it's you see all the fish and all the seaweed and all what we do. So thank you very much, and I think, okay.
It's a pleasure to be here with you. I'm already written what I must say, because I'm trying to don't say what I mustn't say. My name is VIORYL and I am a fundamental pharmacologist. I'm representing AMIROY or AMIROY, but whatever you want, you can call us. We will reply you. And we are it's a company rooted in the belief that the sustainable innovation is not just a possibility, but is a responsibility. So it's the core of our job. As you see here, AMIROY don't just research, is applied also. So we are going to search the new molecules or new resource of biomaterials. We are bring them to the laboratory. We make a new formulation of this biomaterial. Then we do everything to bring this formulation to the market. Behind each of our formulation, it's a great teams that I am a co-founder of this society with my husband, Hamed, and we have the other people that help us. We have a partnership from the agricultures, from the farmers, towards the banks or all the technical institutes. AMIROY is expert in natural biopolymers like chitosanes, lignosulfonates, alginates, cellulose derivatives, and other bio-based materials. And because we are searching to propose a renewable, sustainable applications of new materials. Let's focus on this nice biopolymer. It's chitin that it's found on crustacea, insects, and mushrooms. And we are able to bring it to chitosan by cutting these acetyl groups. And it became a very nice biostimulant, for example, in the agriculture parts. And for the agricultural parts. And we, for example, we have made a patent on the different chitosans that we can bring to each other. This part is how we can bring the different chitosans to each other. It's patented. For example, if we are using this formulation that I've showed you on the grain of wheat, the up panel show that don't receive these molecules, but the below panel show that the same grains with this formulation on the base of chitosan, you will see the different root development. And in the right one, I think, no, it's the left for you, you will see that it shows the antifungal, even antifungal aspects also. I know that we have many scientists here, so we are going to some data, some digits. For example, we have a formulation which name is eliciton. It's working on, it's applied on the leaves of corns, and it's produced in the base of chitosans from shrimps and insects, and it's showed on the field that it can increase resistance to fungal disease above 50%. It's improved nutrition uptake above 30%, and it's make the more resistant systems, so the plants won't go down when there is a great wind above 35%, and the recolt or the harvest would be around 10% more. And all of that is something like water. There is no any chemical components, just the chitosans from shrimps and insects. But in the other hand, AMIROY proposed also all his expertise and facilities to the other industrials or academic partners. We are proposed an A to Z product development process. If you have an idea about, or if you have discovered a nice molecule, a nice future biostimulant, biocontrol, something for the agriculture or other industries, we can propose you our concept of development, then testing and validation if it's in agricultural field with our farmers, or in the institute technique that they are working with us. And we do all the regulatory compliance like homologation, or in French we say autorisation de mise sur le marché. So that means that all the regulatory things that with European committees. And at the end, we are accompanying you to a market launch, means that we are do all the market study and even put this molecule in the market. We are we are very good in the scientific, industrial, and economical part, and I will show you that two examples behind the agriculture parts. Here we are we are asked by another industrial which produced a mulch from hemp waste. This one, a kind of carpet that is used to don't let the other undesirable herbs come out. So they are asked us something biologic, not chemical, to put on this carpet to make it more have a longevity, more longevity, because it's biodegradable and it would be really soon go away. And in the other hand, the formulation that we have proposed make the carrot cultivated on this carpet more in quantity and in quality. Still, it's something like water from chitosan and other molecules. The second example is that new formulation for the treatment of industrial wastewater as an alternative to aluminum-based components. Here we are asked from another industrial which is in the making the papers, and they have the wastewater like that, as you see there, the ponds are like that, and they need to replace this aluminum and have a clear water to put it back to the river. So we are arrived to make this formulation for them also, and they were happy. So I'm thank you for your listening to my speech. I hope we can have a discussion laterly, and I'm really pleased to see you.
I am a biotechnologist and I work for VIORYL. VIORYL is a chemical industry specializing in 4 different domains. We produce industrial fragrances, flavors, agrochemicals, and fine chemicals. And today I will present semiochemicals, which is part of our portfolio for natural products in agriculture. So I'm pretty sure that everyone here is familiar with the term semiochemicals, so I will be very quick with my introduction. Semiochemicals are just natural chemical signals produced by organisms to communicate, and they're divided in 2 main categories. Pheromones, which are signals that are produced for communication within the same species, and they can be sex pheromones, alarm pheromones, trail, territorial aggregation, and the list goes on. Or they can be signals that they are produced for communication between different species, and in this case the term is allochemicals.
Emma,
Yes?
because you have a small voice.
Yeah, I'm sorry. You cannot hear me at the back.
Sorry, yeah.
Is that better? Thank you. Okay, so the use of semiochemicals in agriculture is not a current trend, it's not a new concept. It actually started quite early in the 60s when scientists successfully isolated and chemically synthesized the sex pheromone of the silkworm moth, which marked a major breakthrough in the area of chemical ecology. A few years later, late in the late 60s, early 70s, pheromones began to be used as part of pest monitoring strategies, particularly for Lepidoptera, which is a very important pest that is catastrophic for a big variety of crops. And from the 80s onward, pheromone-based control measures became more widespread and popular in agriculture, not so much as standalone measures, but rather in combination with other measures and integrated pest management strategies. And how can we use pheromones in agriculture? There are 4 main routes that we follow. The first one is for monitoring pest populations. So we use pheromones to detect and track pest activity early, and that allows the farmers to intervene in a timely manner. So that means that they can spray at the start of the infestation with lower doses and spray even restricted areas. In any case, you lower the input of chemical pesticides into the field, which is great. The second route is mass trapping, which is quite self-explanatory. You use pheromones into bait stations, into traps, to lure a large number of adults, insect adults, and thus reducing the population pressure. The third one is mating disruption, where you release pheromones in clouds and you saturate the environment with sex pheromones, which confuses the mates and blocks them from finding each other and reproducing. And the last way, and in my humble opinion, the most efficient one, is in attract and kill strategies, where we use pheromones in combination with an insecticide in bait stations or in traps to attract and eliminate pests selectively. and very efficiently. And in any case, in any route that you decide to follow, the advantages are great, and they're great for the consumer because you can consume residue-free products, for the environment, of course, and for the farmer. And because we're talking about species-specific control, we preserve biodiversity in the ecosystem. We manage to attack pests, and we do not pose any harm to pollinators or beneficial insects in the field. And the last advantage, it is a great alternative control measure when conventional chemical measures are not viable or they fail due to resistance. The insecticide resistance is a major issue for agriculturalists, so pheromone-based solutions is a great alternative. So our company, following the trend, started its journey into the research and development of insect pheromones quite early. It started in the early 80s. And in our company, we have managed to develop and commercialize 3 sex pheromones for insects for 3 very important, agronomically important pests: the olive fruit fly, the banana weevil, and the palm tree weevil. All these pests are very, very notorious to handle, and there are no chemical measures, or the chemical measures available are very restricted, especially in EU. And of course, we have engaged in many research projects studying other pheromones for other insects, which unfortunately didn't end up in a commercial product. And I'm going to discuss the reason why at the end of my presentation. And the most notable milestone was the industrial scale synthesis of the sex pheromone for the olive fruit fly, which led to the development of the eco-trap system. And what is the eco-trap system? The eco-trap system is something very simple yet efficient. It is the system that you see here in the photo. It belongs to the attract and kill strategy. It includes that white capsule there, it includes the sex pheromone for the olive fruit fly. And also in that green envelope, we have a food attractant that attracts even more bigger numbers of flying insects into the trap. And the whole system is covered with deltamethrin, which is a very efficient and effective insecticide that poisons flying insects upon contact. We have managed to register this product, which is the most difficult part of the whole process. It has its own Annex 3 dossier. It has been approved as a plant protection product under the EU regulation 1107. We have tested the product thoroughly. We have conducted field trials in Greece, Italy, and Spain. And it is a great controlled system for organic agriculture. It has been certified by Greek and Spanish authorities alike. And it can, of course, it leads to residue-free products, to better quality products. And it can be successfully combined with olive fly parasitoid release or other biological control measures. And why eco-trap is so important? There are 3 key factors that justify the product success. And I think this is a recipe for success for any other scientific project that wants to come into the market. First of all, the system is designed for a very important cultivation in Greece. We're talking about the olive cultivation, which is massive. It is the top tree crop in Greece, largest in areas and number of trees, of course. It has significant economic impact. It generates 11% of the agricultural income in Greece and represents 0.3% of the national GDP. It is a major key export product, and of course it's a dietary staple for the Greek population. The second key factor is that it targets a very high impact pest. That little fly there can be detrimental for the production of olive oil. It can cause up to 30% production loss, and of course deteriorates the quality of the products, both the olives and the produced olive oil, significantly. And the third key factor is that it came to fill a gap, and that gap was that the traditional control measures that they are available to fight that pest are very heavily restricted in EU. We have, of course, some registered chemical active substances, which you can use only in a specific way, in specific doses, And now with the introduction of the Green Deal, I think most of those active substances will be even more restricted and in some cases banned by 2030. And of course we have pyrethroids, which is, we all know about the resistant concerns. It's almost no efficient now for tackling the olive fruit fly. And yes, okay, there are some biological control measures, but they're not very efficient when we have high pest pressure. And they have to be used very carefully and only when environmental conditions are favorable. So if pheromone-based solutions are so efficient, so beneficial, and so green, why there are no more plant protection products based on chemical ecology principles? And the reason is dual. The first one, of course, for the business is the high development cost. It is super expensive and it's super time-consuming to isolate, study, synthesize in an industrial scale an insect pheromone and to test it. And the most important one is the regulatory hurdle. Pheromones are considered by EU as a new active substance. So you have to go through the same process that you go for a chemical active substance. And I'm going to give you just some numbers to see that for a company, it might cost 2 to 3 million to approve a plant-based product, a plant protection product based on a pheromone, and it will take at least 5 years to prepare a dossier and to submit it for approval. And this cost and time that I'm giving you is just for the registration. I'm not talking about the time and cost that we need to calculate for the research and the development and the trials. So it's not a very financially feasible thing to do for a company. And on that rather negative note, I'm going to wrap up my presentation. And I would say, personally, I found chemical ecology a very fascinating field, and it offers very promising tools for agriculturalists. But if we don't overcome those regulatory constraints, all the results will stay in the lab, and they will never reach the field. So it was rather quick, so thank you very much.
So it's a pleasure to be here. As I will explain, it's a bit different environment for me. But anyway, I'm very happy to be here, and I really think that we can kind of cross bridges between the different sectors, mainly the seaweed more applied research and industry, and with chemical ecology. So I'll just give a brief overview. So who am I, just to place you? So I'm a marine biologist as formation, phycologist. Trained in the Azores, started with the seaweed taxonomy and ecology, and then went into seaweed cultivation and applications. Some of my, I would say 2 of my major achievements was to co-found the company, Portuguese company Alga Plus in 2011, which is a seaweed farming company that farms seaweed integrated with fish in Aveiro lagoon. And the seaweed is used for food, cosmetics, and other high-value applications. So we were quite pioneer in 2011, and the company is still thriving, and I hope that it thrives for many, many more years. And recently, when I was also involved, I'm still involved in International Seaweed Association. So for the ones that don't know, it's an association that exists officially since 1970. But in fact, it's been organizing the International Seaweed Symposium since 1952. It's the oldest seaweed symposium. It happens every 3 years, and I invite you, anyone that is interested in seaweed, either from the ecology up to industry and applications, to attend that symposium. Next one will be in Qingdao in China in 2028. Anyway. Now I have, just to say, I have a consultancy company, and I'm here kind of representing that. Really matchmaking, as I say, I'm a bit of the seaweed Tinder, matchmaking producers with end users and researchers. And I also have a part-time at the Bantry Marine Research Station with Julie, where I actually started working after my postdoc, well, doing my postdoc in 2010, long time ago. Anyway, and that's my favorite seaweed. If you talk with a seaweed person, they always have a favorite seaweed. So mine is Plocamium cartilagineum, which I love. It's a red, beautiful seaweed, full of also defense mechanisms. So my why in seaweed is actually in the sector is really to foster seaweed for food. This is what I love. It's my passion. So basically democratizing production and application and integration in the food value chain. That's my main thing. And also a disclosure note, I'm not a chemist. I work with chemists sometimes, but most of my research has been focused on seaweed cultivation, the impact of environmental factors for optimizing seaweed biomass production, yield, but also composition. But mostly going from the lab to an aquaculture set, commercial scale, so very hands-on, very operational, so a bit different. Everything that you see there related with chemistry, biochemistry, it's always in partnerships. It has always been in partnerships with a lot of colleagues across Europe. So, but anyway. So brief overview in seaweed, I'll go very quickly. So just to show you that there's a lot of generalization sometimes, and I like to show this because in fact there are over 10,000 species and seaweeds can be very different. They can fit in your hand or even smaller than that, and they can be much bigger than you. This is an example also of Saccharina latissima, which is a seaweed that also exists in Europe, but when it's growing in Canada, it reaches much bigger sizes than when we grow it, for instance, in Ireland. So just to show you there's a lot of diversity in species, and that of course affects everything from production systems to the composition and of course their value. There are 36 million tons produced in the world. I like to bring these numbers because sometimes people think this is a new thing, seaweed, and it's not. It's been here for a long, long time, for millennia, and it's a very important worldwide industry. Most seaweeds, despite we have 10,000, there's only around 5 to 6 genera or types of seaweed that are produced worldwide, mostly in Southeast Asia. And you have them here. Biggest producers are Indonesia, China. They're always kind of changing one year to the other. But these are the main producers of seaweed. And most of the biomass is going for food, actually, 75%. Either to be used for polysaccharide extraction, agar, carginian, and alginate. That's the biggest, the bulk biomass goes towards that. But then also for consumption of sea vegetables. And then recently biomaterials, biostimulants, etc. Many other applications that I won't go through that because it will take us all afternoon. So brief overview of the production system. So we can grow seaweed in different ways. We call it the low-tech cultivation systems. These are mostly done for species that can grow vegetatively. So like grass, you cut them and they keep on growing. So it's a very easy system to upscale. Only a few species can have these features, but it's actually most of the biomass is produced this way, and in some of the examples that you can see there. What we have in these intensive systems, and just to give some introductions in relation already to chemical ecology, is because intensification, like it happened in agriculture, is bringing diseases, is bringing pests, is bringing a lot of issues for these farmers. So more and more, besides studying and optimizing infrastructures, there's a lot of work and research being done at the chemistry of the seaweeds and how it can be improved also towards increasing the defenses, mostly against the parasites, besides the genetic approach. Another way that growing also at sea, let's call it, but a more high-tech system, and these are some examples that can around Europe, but also the nori production in Asia, is these systems, they always require a hatchery, normally very specialized personnel, and there's other things that can be done here, like more input into strain selection, and also to some customization in terms of the biomass. So I won't go through all the details. If you have questions, then you can ask me. But that's mostly kelp that you see there in Europe, and then some nori in Asia. And then you also have land-based systems. Of course, we are increasing technicality, if we want to call it like this. This is mostly done for two reasons. Either for species that are small enough that they would not be able to be upscaled at sea, so that either they would be lost, or yeah, mostly that you would lose all the biomass, all the investment that you have put in, but also for allowing for customization of more fine cultivation protocols that will induce higher value chemical composition of the seaweed itself. And these are some examples. This is in Canada. It's the largest land-based facility, being around Halifax. They are today, they started 40 years ago. They are today the biggest one in the world, up to some very niche production that you see there in the middle, those photobioreactors with pink. It's a coralline algae that's in Codif, Fitormer, in Normandy, in France, that they grow it. So it's a cosmetic company. They do the production themselves in order to control everything, all the process, and the quality mainly of the impact. And then you have different examples today. All of these systems can be also integrated with with animal aquaculture. So, and also plants, but also more animal aquaculture, either from fish or shellfish. And this brings advantages, but also challenges in terms of how will the different trophic levels will coexist in the same space. But the biggest advantage of IMTA, it's a much more sustainable, not only environmentally, but also economically, way of upscaling seaweed biomass production. And this is also possible in all of the systems I mentioned before. This is just to say that, of course, you need to, it's not a recipe, so you always need to choose the species, depends on what you want them to. So Europe, where are we at this? So Europe really wants to become a big player in the seaweed world. So the target is to reach 8 million tons in 2050. And I will show you next where we are today. But a lot of SMEs driving the sector, a lot of companies coming out, it's all in the news. And there's a lot of funds from the EU towards now the algae, microalgae and seaweed also. So there are some key challenges, and I think that there's, of course, we all know ways of cooperation and cost actions of other projects. So where we are today, more or less around 800 tons. So it's still a long way to go to the 8 million that Europe wants to see. But I mean, we are moving there, and we are keeping the optimism. And of course, this needs a lot of collaboration. Not only within the seaweed sector, but I think most especially cross-sectorial collaboration. This is some of the examples of the species that are being produced that I will focus later on in a bit. So how much is the time I have?
You kind of gone over.
Already? It's 10 minutes? Is it 10 minutes only? Oh, I thought it was 15, sorry. I'll go quick. So chemical ecology, I'm sorry about this. So we are early, right? So I am using the time. Sorry, my bad. So what about chemical ecology in seaweed? So in fact, when we look and I'm coming from ecology and as a marine biologist, as a farmer, and I won't go through all of the details, and you have the presentation afterwards, so I think it can be shared for everyone. But for me, the most important thing is when we wanted to start, so we always need to look to the shore, to the wild populations of the species, and then learn from nature to go and build up our company, our business model, right? And especially when we are looking at seaweed species, we need to choose the species, of course, that they have a market demand, because otherwise it will be very much to be profitable. But very importantly, when you are looking at the set of species on the type of species, we have we do a SWOT analysis. And one of the things that exists in the SWOT analysis is how clean is this seaweed when I go to the shore? And the seaweed's biomass, the species, they keep themselves clean because of the defense mechanisms that they have towards epiphytes, can be other seaweed, bacteria, but mostly grazers. So when we look at the species that today we are farming and that we are able to upscale, many of them have this ability, and these are the compounds that then we are exploring as bioactive compounds later on. And all of these can be modulated. Of course, it needs much more research still today to really understand the mechanisms, but it is part of the basic knowledge to when we are starting our seaweed farm business. And there's a lot of literature around it. And there are things, just small things that I like to make more relevant, or because they're kind of new. And one of them is, for instance, looking at the chemical composition, mostly glycolipids, and understand where is the seaweed coming from. Not only wild populations, but also the farms. So we are already looking at traceability tools from what the glycolipids can tell us in seaweed and be able to trace them. So there's a lot of cool things happening. So, and I won't go through this. In production, cultivation, chemical ecology is crucial. We have a lot of information which, with types of the compounds that we should kind of understand and modulate, and if possible add even to some of the phases of our cultivation steps in order to get the best yields. And this happens not only from the hatchery stage that we are controlling, as I mentioned, from the beginning, so our life cycle procedure, so we want to trigger the change of phases, but we also want to customize the chemical biomass right at that stage, up to the processing phase that we are looking at timings, we are looking at the types of processing that will affect a lot of these bioactive compounds. So always chemistry, of course, and things that we can modulate in the company to develop. So of course there's bad things, and we look, seaweeds are bad for corals, I want to say that, but seaweeds are good for whales. This is all over the news yesterday, just, I don't know if you saw this, but there's orcas that use seaweed to scratch themselves, to improve their skin health. Yeah, so this is just a new research. So it's quite cool, all of the different things that you can do mostly. I showed just some case studies on kelp, that kelp, for instance, I don't know if you're aware, depending on the time they're harvested, we will look at different chemical composition. So we can plan if we want to go either for nutrition purposes, or if we want to go for biomaterials, we can choose the time of harvest, and that because we will have changes in the carbohydrates and phenolic compounds, etc. But also there's a lot of studies looking at how the seaweed itself is defending itself from grazers on the long line. So there's a lot of things on this. On the ulva, and this is just to show you, there's been two cost actions already that I was involved in the recent years looking at ulva biomass. One is the phycomorph that was finished. Another one is still going on, which is seaweed. And in all, especially phycomorph, we did a lot of nice work with the morphogenesis and looking at the bacteria for the microbiome and understanding how changes in the microbiome at the aquaculture levels were changing, for instance, reproduction traits. I don't know if you're aware, but ulva, the sea lettuce, when it's stressed in terms of nitrogen and light, for instance, intensity, will immediately trigger reproduction. And a company can lose all the production in a couple of weeks. And looking at the bacteria, we were able to see that the fact that we had the good bacteria in the water culture were able to prevent that triggering of the reproduction. So there's a lot of nice things happening, and that the companies are now working. Asparagopsis, of course, that we all have heard of, that we have the bromoform, that it's good, but it can also be very bad, and it's actually puts a lot of questions regarding, is it safe, this seaweed, not only for the use, but also very important for the operators. Just to give you a personal example, we were drying this species that creates blooms here in Peniche area. And when we were drying it, the operators couldn't be in that room where we normally dry with the dehumidifier other species, because the levels of the volatiles that were released were so strong that it would affect eyesight, breathing. So there's a lot of things to say about these species when we are looking at it. And then porphyra, very crucial. I won't go there, I don't have time. But anyway, we have all of this I can share with you later, you can ask me. And as you see before, it is important that if we know how to control these chemical traits, not only in the production, but also in terms of the value that we want, we can really tackle in a more fine way the business model of the companies. And a lot of us are looking into this and working with you guys on how to improve our productions. So some key takeaways, and just I like to make people connect. So I just put here 4 people that I think that should be in this cost action, because they are working with the seaweed, but also looking at chemical ecology and how it could be triggered. And there's people from Germany, France, Ireland, and Portugal. Yeah, and that's it basically. And thank you, everyone. And I'm sorry for taking so long.